Thank you so much. Thank you for to the organizers for inviting me today. This is pretty exciting to be able to talk about a field that we love. So I just actually want to get an idea. If you feel comfortable answering this question, please raise your hand. Who here has either met a genetic counselor before today, or knows a genetic counselor? Yeah, a show of hands, please. Okay, yeah. There aren't a lot of us out there, as evidenced by Lauren's talk. Fewer than 5,000 genetic counselors out there. And one of the things that we'll probably illustrate in the next two talks is a little bit about how niche our roles are, and at the same time, how much our roles are expanding, as evidenced by Lauren's talk earlier, and just all of the different practice areas and research and non-clinical roles that genetic counselors play. So we are a growing field, and we're going to just take a deeper dive into that niche area that we live in, which really does interface with so many other disciplines. Be they social work, nursing, certainly hard science research as well, and Lacey will definitely be talking a little bit more of the data-driven efforts that we do. So as Melanie has already touched upon, as assistant program director, I'm involved in teaching the genetic counseling students. I draw from our library resources when it comes to audiovisual learning, and certainly background reading for teaching the Intro to Genetic Counseling courses. I'm involved in admissions as well, and student research advising. As a cancer genetic counselor, my involvement in research is a little bit different. I'm primarily involved in patient care, so that's really where I take a shift and think about, okay, what does it mean to be a patient? What is the impact of genetic disease on a patient? What is the impact of illness on a patient? And so many of the things that we are curious about stems from our experiences in patient care. So much of our scholarship and writing derives from either gathering data from quantitative or qualitative research methodology, or writing about our collective observational experience on patient care. So genetic counselors broadly serve many different roles on research teams, but here's just a few highlights. Much of the projects that I will be discussing today, many of the projects will be genetic counselor initiated. Again, answering the question, what am I curious about when I'm thinking about how to best care for my patient population as a whole and my individual patients? Recruiting for a basic science project, one of the earliest projects that I worked on as a cancer genetic counselor, really was recruitment. And because this was circa 2002, and so many of the genes that we have clinically available for genetic testing in the hereditary cancer setting, available today, we didn't know about back in 2002. So many of our research projects were gathering families that have multiple cancers in the family. They may go through multiple generations with similar cancers, such as colon and stomach cancers can go in some families. Breast and ovarian cancer can go in, together in some families, or breast and uterine cancer. So we were collecting families of interest, hoping that someday we'd be able to ask the question, what genes could be a contributing factor to these family histories, in the hopes that by identifying a risk gene or a mutation in a gene, we could also offer genetic testing to other family members so that they can better understand their lifetime risk of developing cancer and proactively pursue screening for early detection, for example. Also, knowing the gene that may impact a patient's family history, or responsible for family history, can help us, hopefully down the line, develop therapeutics that either treat the cancer or, even more so, proactively reduce their risk of cancer. So we call that chemoprevention. So those are some of the projects that we were involved in very early on. And then also, very often we're involved in huge, large studies that are maybe whole exome sequencing of a large cohort of families, and maybe adding sub-projects to that larger sequencing. Maybe like, let's assess our consenting process for consenting parents of newborns who are considering whole exome sequencing. You know, the consenting process is really complex. What questions can we ask about this process to help us better understand the impact of the consenting process, whether the parents have a good understanding of the consent process as well, and whether there's any decisional regret perhaps after embarking on whole exome sequencing for their newborn. So topic expertise for projects, we are often called upon to add our expertise in a larger project that may not necessarily be straight up involving human genetics necessarily, but because we live and breathe in the area of thinking about patient first, thinking about informed consent. Many genetic counselors are members of the institutional review board at their institution, and also are brought in, even in large pharmaceutical companies, are brought in as kind of that expert who understands the patient experience. So the genetic counselors are involved in that area as well and offer their expertise in that area as well. So if I'm illustrating kind of a team approach to much of what we do, that is quite intentional. So as advisors, then we also serve as primary advisors for future genetic counselors in their master's thesis projects, and I'll be presenting a couple of those today. So what kind of questions do we ask? I'm just going to take a little bit of pause here. So much of what we do definitely stems from patient care. Our history, as Lauren pointed out, comes from understanding Mendelian inheritance. So Huntington's disease and Tay-Sachs disease, these were some of the longer standing diseases for which we understood the inheritance pattern and therefore have more of a sense over the history of our profession of what patient care and what's involved in patient care, especially when it comes to the implications for family members, not just the person for whom has the disease, but also their parents, their siblings, their children or future children. A genetic condition impacts a person and their family in multiple different ways, okay? So with that, genetic counselors often ask questions along the lines of patient care, patient understanding of their genetic disease, what informational gaps are there, what kind of resources do we need to provide that is readily available? And as you probably know and appreciate, there's a lot of information out there, but not all of the information is curated to the degree that one would want, is in terms of making sure that it's easily accessible, but also accurate and up to date, right? So those are some of the kind of questions that we might pursue as genetic counselors. But the psychosocial impact of genetic counseling, genetic testing, and the impact of illness on a patient and a family are also areas. And so just to broadly describe that as the psychosocial care of patients. And you can subcategorize that across the lifespan, okay? An individual may meet with a genetic counselor at different points in their life. It could be in the newborn period when a child is born with multiple congenital anomalies. It could be in the prenatal period, certainly, in which case the counseling is of the prospective parents. Also childhood, maybe a very typical scenario in pediatrics is a 7-year-old with pervasive developmental delay would be a typical referral to different disciplines, but genetic genetics is often one that a pediatrician will maybe at the outset refer to a genetic counselor or a geneticist, or maybe much further down the line. And by then, if you can imagine, many parents and families having gone through multiple specialists searching for a diagnosis that might explain their child's constellation of features, probably feel a good amount of fatigue during their diagnostic odyssey. So part of what we do is we understand, we support, we help them through that diagnostic odyssey through information, accurate information, but also just supporting and recognizing and naming some of the feelings that they're coming in with in the clinic. We are the 50th person that they've met regarding their child, because they've probably not only met with health professionals, but social workers and also developmental specialists, neuropsychologists, etc. So some of our scholarship is in those areas, and our writing is in those areas as well, to help broaden the understanding of the patient experience. And then extending further into the lifespan, many of my patients have von Hippel-Lindau disease, which can span the lifespan in terms of risk for tumors as young as childhood, with increased risk for eye tumors. So children do have genetic testing at a pretty young age. So as they're emerging from their childhood and transitioning into adulthood, not only does that time have is a dynamic time for maybe exploration of career, exploration of relationships, and for the parents themselves launching their children in that time period. It's a pretty critical time period. Having a genetic diagnosis and taking on your own medical care is another dimension to emerging as a young adult with von Hippel-Lindau disease. So I'll talk a little bit more about that data as well. And then you are also thinking about perhaps adults of childbearing age, maybe decision-making regarding if they know they have a family history of cystic fibrosis, then they may be thinking about carrier testing prior to establishing a pregnancy. And then further on into the lifespan, genetic testing for inherited cardiac diseases, certainly Huntington's disease and inherited cancers really do extend further beyond there. So many of the questions that we ask do need to touch upon different time points in a person's life. So much of the genetic counseling driven research tend to be quantitative or qualitative, and surveys are certainly a very convenient way to ask the same set of questions, closed-ended questions, to a broader audience, whether it's a broad audience of genetic counselors to understand their current practice, or to survey a specific patient population to understand the impact of their disease. Qualitative studies often are interview-based. They can be focus groups as well. And we ask open-ended questions, and often provides a richer set of data for us in helping us understand it. It's also very amenable to a pilot study to better generate a hypothesis for further follow-up with quantitative studies. And so examples of genetic counselor surveys are ones that we might be identifying current trends in clinical practice. For example, if new technology or new testing becomes available, then how are genetic counselors consenting for those? How widespread are these new tests being offered? What are some of their hesitations about offering it, or what are some of the drivers for offering the test? Very often it is patient demand. But again, as genetic counselors, we are somewhat gatekeepers of information, but we don't consider ourselves like the traffic cop. And so, you know, we do understand that patients seek genetic information, and what we really strive to do is provide kind of a balanced overview of the impact of genetic testing and the potentials for genetic testing, but the limitations of genetic testing as well. And sometimes we may use genetic counseling surveys to identify gaps in our own knowledge. You know, what are some of the areas that we haven't really explored before? And genetic counseling as a profession certainly impacts the person themselves, right? So caring for others, at the same time, you also need to make sure you're caring for yourself. So there are multiple studies out there that look at compassion fatigue and self-care. Psychosocial care of patients is an area that is very amenable to genetic counselor survey, and much of our literature is actually grounded in the psychosocial care of inpatient care. So here's an example of a genetic counselor survey that was conducted in 2017-2018 by my student Elizabeth Sheehan. She was very interested in understanding current practice for representing individuals who identify as transgender or gender fluid or on the gender non-binary and gender non-conforming. And so just a little bit of background of how we draw a pedigree. We are all trained in standard nomenclature. That was our most recent publication of standard nomenclature was in 2008, so 10 years ago, okay? Things change in 10 years. So just to provide a little bit of a background. Okay, so a circle in this pedigree represents female, and this arrow represents the individual for whom it's presenting to the clinic, so the proband. And this relationship line is a sibling line right here. So this is a 45-year-old female who has a older brother who's 51. Down here is a descendants line, and so he has a child, a son, who was conceived with a donor egg. Okay, so that's just to represent a donor egg. And you'll see that there is no line of relationship between the brother and the anonymous donor. And then down back to our proband here, she has 4 children, a daughter, twin sons, and here we represent her adopted son as well as a member of the family, but genetically or biologically not related to her. Now, up here we have the relationship line to her parents. Her father is 72 years old, her mother is 74 years old, and these are her grandparents over here. And there's her father's parents, so her paternal grandparents. And we include ancestry up top in the pedigree. Over here, the diamond can symbolize multiple things. So when you're obtaining a family history, and perhaps you might not have asked the gender of the family member, you can use a diamond to represent gender unknown. In the 2008 publication of standard nomenclature, there was also a conversation within the genetic counseling work group on how to represent patients who identify as transgender. The recommendation was to use a diamond and then underneath write transgender male or transgender female. So Elizabeth, my student, was interested in finding out how consistent are we being across the profession in using this symbol. It's kind of like a one line in a paper that we're all familiar with. And does it cause confusion? Because it's not readily obvious the gender assigned at birth when you see a diamond. And here also, I also want to explain that from the risk assessment point of view, perhaps this is a woman who is presenting to the clinic for evaluation of her cardiac family history, and she discloses that her transgender brother was diagnosed with long QT syndrome. It's not readily available, obvious what his gender was assigned at birth. So the potential for that risk assessment confusion exists when we are not consistent in our clinical practice. And so with that, she surveyed the genetic counseling profession through the NSGC listserv, and with this aim of identifying current practice in pedigree nomenclature, assessing genetic counselors' confidence in their ability to address the psychosocial needs of their transgender and gender non-binary, gender non-conforming clients, also to establish educational needs and regarding gender identity and fluidity. And through this survey, through the National Society of Genetic Counselors, she gathered quantitative data, which was analyzed via SPSS, and also it was a mixed method survey, so she had some open response questions as well to ask respondents to elaborate on their choice of pedigree symbol, or whether, if they did not choose a ideal pedigree symbol, what were some of the things that they would like to see in the future. And so she analyzed that with thematic analysis. And so she had a really, for genetic counseling surveys, 469 responses is a pretty decent number. It probably only represents about 20% of the membership, and included both practicing genetic counselors in non-clinical areas, as well as genetic counseling students. And there's certainly a lack of agreement on pedigree nomenclature for transgender and gender non-conforming individuals. So just to serve as an example, a piece of her data, if you were to look at the NSGC 2008 recommendation, the recommendation is to use a diamond. And the National Comprehensive Cancer Network also came out with recommendations, and their recommendation to represent a transgender male was to have a box with a circle in the middle. And then we also, this was a multiple choice question, and provided a few other choices, and ones that were more gender identity affirming, including a box with XX to represent the female karyotype, or trans male underneath, were considered the more favorable option. But many did, 30% did choose the symbol consistent with NCCN, 60% chose this one or this one, and then very few liked the diamond. Also included in her survey was an attempt to assess, genetic counselors' point of view as well on symbols, and pretty widespread agreement that symbols need to be updated. We need to engage stakeholders to better understand what would be most appropriate, so that we can implement that and have it consistent across the profession. But the one thing that very much wanted is gender affirming symbols that appropriately represented our patients. And additional education in psychosocial care and medical care for transgender and gender nonconforming individuals was also widespread. 96% of the surveyed population desired more education. So moving to patient-related surveys, very often they can be asking the question, what are patient preferences for the approach in medical care? Identify informational needs, knowledge gap, and resources that they might need. Adjustment and coping to a genetic diagnosis or an illness, psychosocial needs, and in both of those, we often look for validated surveys, validated instruments, and so very often we will need to call upon our science librarians to help us kind of better understand. So I know nothing about the social science literature or this medical sociology literature. Can you help me understand or look for maybe previously validated instruments that might be useful for the question that I'm asking. And the impact of testing on clinical management is often certainly one of those kind of the before and after. Before you had genetic testing, what were some of your medical management concerns and medical management approach? After genetic testing, what changed would be a typical question. So moving to qualitative studies, looking at genetic counselors' approaches to patient care. Katie Schwarting did her research study in 2017 by inviting 10 genetic counselors to a phone interview, which was recorded and transcribed and analyzed for themes. Her interest was to better understand how genetic counselors who work in Catholic hospitals handle religious directives within their patient care. Her findings was that genetic counselors reported that navigating religious directives and specialist referrals were part of their role. It certainly impacted how they provided patient care and what they discussed with regards to pregnancy termination. They knew who they could refer their patients to if their patients needed additional information. And overall, genetic counselors felt that they were able to support their patients appropriately. Another qualitative study by Lindsay Schmidt was on certainly the young adult perspective with von Hippel-Lindau disease. So I kind of queued you up for this part earlier in my talk. But certainly, von Hippel-Lindau disease can, again, impact a patient from the pediatric years with young onset benign tumors that need to be treated quickly. So, for example, eye tumors, you definitely need to remove them before it impacts vision, or use laser or cryoablation to remove them before it impacts a child's vision. They are also at risk for benign tumors of the adrenal gland, which can play with their blood pressure, and with persistent elevation of blood pressure that can cause secondary effects like cardiac effects on the heart, for example, or effects on the kidney. So we need to know if a child who is at higher than average risk for these kind of tumors, in order to screen them appropriately, dilate their eyes, look at their retina on a yearly basis, and also do blood work and measure their blood pressure on at least a yearly basis to screen them for pheochromocytomas. And then also they are at risk for benign brain tumors of the cerebellum, and therefore by age 9 they need to have a baseline MRI of their brain and their spine as well. Okay, so impact-wise, you can imagine why there would be an inspiration to write a book. Myself and a few of my colleagues in the VHL Alliance wrote a book for children to help them navigate, and their parents to navigate through the common questions that are very much on the practical level. You know, like, what is an MRI? How is it going to make me feel? And what is that experience like? So that book was also illustrated and contributed by children who were willing to take photographs of themselves entering an MRI machine or getting ready for a blood draw, and also drawings that they provided as well. Moving into the young adult age group, however, is again a pretty dynamic time where they are often taking on more of their care. So Lindsay conducted 10 interviews, 5 were female, 5 were male, and or young adults who were diagnosed with VHL at least in their teenage years, if not earlier. And the aims was to understand the psychosocial challenges of young adults with von Hippel-Lindau disease. And overall, in her study, she found that respondents have a remarkable degree of resilience. Overall, they say, you know, I have more challenges than my peers of the same age. At the same time, very much one of the emerging theme that she found was that they often compare themselves to other situations, friends in other situations, whom they deem is more difficult and would be more challenging if they had, let's say, a mental illness, for example. So, so their sense of their adjustment and coping to their own disease is actually quite remarkable. It does have a polarizing effect on relationships that friendships whom they thought were strong before, after becoming symptomatic with VHL, with their first tumor necessitating their first surgery. Then, I think I'm practically quoting one of the respondents, you kind of learn who your true friends are, and friends who you did not realize were that close to you suddenly come up and kind of take on the role of support. Additionally, it does impact their decisions about having children of their own. Many consider adoption, and many consider not having children as well. So resources that we often ensure that our students are aware of course, is Brandeis Scholar, and PubMed, PsycInfo. We often might do some searching in there as well, and our librarians definitely provide so much guidance, and kind of like, I can't come up with the search terms, please help. And then our institutional repository for looking at past thesis work and seeing what can we build on that, how can we build on that. So I'm just going to end off on one case report. So other examples of scholarship within the genetic counseling profession involves reporting on cases that are instructive. So I work at Mass General, and given that one case record from the Massachusetts General Hospital is selected each week as an instructive case to publish in the New England Journal, sometimes they are genetics-related cases that may be so prototypical of what one should look for and exemplary of what one should look for when making a genetic diagnosis, or actually have such an interesting pathology that warrant publication in order for there to have a kind of an instructive impact on the medical profession. And so one case that we wrote up in Familial Cancer is on a young child, a 14-year-old girl who presented to her pediatrician completely asymptomatic, and she had no signs, no symptoms. She presented for her 14-year-old well visit, and her pediatrician did her usual routine exam, and that included feeling her abdomen, and felt that on one side her abdomen felt distended and firm. So she sent her for a renal ultrasound, and lo and behold, she had a very large 20-centimeter tumor. And 20 centimeters is pretty large given the size of the kidney itself. So that accelerated multiple visits in a very short time frame. Okay, so we're taking a well visit, and now we're escalating it into a medical visit pretty quickly for a 14-year-old who's in high school, or in her freshman year, actually. So she presented to Mass General for evaluation with the pediatric oncology group, quickly went through surgery, and she and her family were referred to us for genetic evaluation just based on her very young age of diagnosis. The average age of kidney cancer across the general population in the US is 67. Okay, so 14 is very young. One of the things that we offered was certainly genetic testing, with the caveat that we do want to help the parents and the adolescents in a age-appropriate way, understand that genetic testing may provide some explanation for her cancer if we find something, but more likely than not, it wasn't going to find anything. And to help them navigate those questions, is this information that would be helpful to you now, or is this something that you want to do in the future? Because one thing is for sure, she is going to be monitored very closely because of her history of kidney tumor. But she presented to the clinic with her parents and her younger brother. And collectively as a family, because they're a very strong family, tight-knit family, collectively decided that they all wanted the genetic information. That in itself is a little bit unique, actually. So just a little bit about their genetic testing, which did actually find a pathogenic mutation in the Birt-Hod-Dubé gene. Birt-Hod-Dubé has benign tumors of the hair follicle. You can see lung cysts or blebs. These are not fluid-filled cysts, but these are kind of air-filled pockets in the lungs, which lead to a susceptibility to spontaneous lung collapse. Not everybody gets the skin tumors, and not everybody has a spontaneous lung collapse in their lifetime if they have Birt-Hod-Dubé syndrome. With kidneys in kidney cancers and kidney tumors can present in about 34% of individuals, so over 60% of individuals never get a kidney cancer. And the age range is based on one larger study, which isn't that large because this is a pretty rare syndrome. Age range can be 31 to 71 with a median age of kidney cancer at 48. And very often, if a person does develop one kidney tumor, they may develop more over their lifetime. But it's a pretty highly variable disease, and we don't usually offer genetic testing for Birt-Hod-Dubé syndrome in a young adolescent who has no personal history of lung collapse or of kidney cancer. So just, again, to take a pedigree to draw out her family, there she is. This is her brother, and this is her father, who's 39, no history of cancer. Her mother's 34, healthy. Her maternal grandmother had a history of breast cancer, and her paternal grandmother had a history of stomach cancer. No family history of kidney cancer that they're aware of. So the impact certainly is that our typical age of offering genetic testing tends to be about 18 or older, because we think of Birt-Hod-Dubé syndrome as an adult onset disease. What do we do in this scenario when we know that she has a genetic diagnosis, she has a mutation in the Birt-Hod-Dubé gene? It has implications for her parents and implications certainly for her brother. Okay, so I'll let you kind of think about some of those implications, but in the long and the short of it is that the reason why we needed to publish it is that her father actually does carry the gene mutation, and therefore he is being proactively screened for kidney cancer. But again, this goes to show how variable it can be in some families. And therefore we have to think about the age of testing and screening for kidney cancer thoughtfully. So web resources that we often go to in the clinical setting certainly are the publicly available databases such as ClinVar and Nomad, and I'm going to leave that for Lacey to dive further into as well. But I just want to acknowledge my colleague Cassie Buck for some of the inspiration of these slides. She is our research advisor for our genetic counseling program, and then the alums for whom the data are presented today. Some of the web resources and also my contact information. Thank you so much.
Rounding out our session today, Lacey Smith is also a Brandeis grad of the Genetic Counseling Program, class of 2014. She took her degree and went straight to Boston Children's Hospital, their epilepsy genetics program. She provides genetic counseling to families seen in the epilepsy genetics clinical consultation program. In addition to her clinical work, Lacey also coordinates the PCDH19 Epilepsy Patient Registry, which hopes to more fully define PCDH19 phenotype. She is also involved in a variety of collaborative research projects that aim to better understand the genetic contributions to epilepsy. Lacey is a member of EpiGC, which is an international network of genetic counselors who specialize in epilepsy genetics and who are dedicated to promoting access to quality genetic services for patients and families affected by epilepsy. Take it away, Lacey.
Great. Thank you so much. Okay. Great. So thank you so much for inviting me to speak here today. You know, one of the really great things about the increase in number of genetic counselors and the expanding roles is the emergence of these kind of not only specialized genetic counselors, but really the subspecialty genetic counselors. So Lauren gave a great overview of kind of how the role has expanded from just, you know, when the genetic counseling field first started, you had the cancer genetic counselors, the pediatric genetic counselor, the prenatal genetic counselor. Then you started seeing the cardiac genetic counselor, the neurogenetic genetic counselor. But really, it's expanding even more to within even neurogenetics. You have even subspecialties. So I see patients in clinic and do research solely in epilepsy. So even over at Boston Children's Hospital, and we see these in the large institutions, we have people in subspecialties. So even in neurology, we have 8 genetic counselors. 3 of our genetic counselors are just in epilepsy. So which is a great opportunity for patients and families to really see providers who are really specialists in their child's condition. So what I'm going to talk to you about today is I'm just going to give you guys just an overview of epilepsy and what is known, and a lot about what isn't known about the genetic contributions to epilepsy. Some of the current approaches to gene discovery in epilepsy and how we're trying to fill the gaps in our knowledge. Sprinkled in there is going to be some helpful resources that we utilize, and I'll pass along to you guys as well. So epilepsy is quite common. It affects about 1 in 26 individuals, and there are about 65 million people worldwide who have epilepsy. So what is epilepsy? Epilepsy itself is simply defined as having 2 or more unprovoked seizures greater than 24 hours apart. So what is a seizure? At least when I first started in an epilepsy clinic, I envisioned a seizure as just the seizure type, as the convulsive seizure, as somebody falls to the ground and has the typical generalized tonic-clonic seizure where they have the convulsive seizure. But really a seizure can be any one of the things that we see here. So it can be the convulsive seizure that we typically see, but it can also be something more subtle. It could be something as a unilateral movement. It could be a visualization or a hallucination. It could be an unusual taste in the mouth. So these things are all considered seizures. But basically what it is, it's an abnormally synchronized event in the brain that may or may not lead to some sort of outward clinical manifestation. So when we're thinking about diagnosing a patient with epilepsy, we're thinking about, does the patient just have epilepsy, or do they have something else? And I don't mean to think about this as, oh, is it just epilepsy? Not in the way to belittle anybody who has epilepsy, because there are certainly, it's a patient who has epilepsy in the setting of normal development, for example. It's certainly something that is disruptive to quality of life, and there's certainly a lot of social stigma surrounding epilepsy. But in the epilepsy clinic, we're thinking about, Is there epilepsy in the setting of normal development? Or is there epilepsy and something else going on for this patient? And the other things that we think about, is there other comorbidities going on? Is it epilepsy in the setting of developmental delay, developmental regression or stagnation? Epilepsy in the setting of autism spectrum disorder? Is there a family history of epilepsy? So is there something else also going on as well? We also think about the presence of epilepsy and also something called an epileptic encephalopathy, whereby looking at the EEG, so when we actually map the brain patterns, is the background brain pattern abnormal? So you can have epilepsy and have a normal brain pattern, or you can have epilepsy and an abnormal brain pattern, and that's a characteristic of an underlying abnormal brain activity. Just thinking about what we know about what causes epilepsy, for a long time, until just recently, a large part of what causes epilepsy was unknown. There was, the obvious answers of brain tumors, traumas, birth injury that can lead to epilepsy, but there was a large portion of epilepsy that wasn't known, so it was considered idiopathic or of unknown cause. Over time, there was a presumed genetic component to epilepsy. So there was this emerging evidence for the role of, could genetics play a part in epilepsy? So there were, the traditional genetic studies that I'm sure you've all read about. So there were the twin studies where they found that there were more presence of epilepsy being present in monozygotic versus dizygotic twins, so identical versus fraternal twins. There was a higher, just empirical data from family studies showing that there was a higher chance for an individual to have epilepsy when there was already a sibling, for example, who had epilepsy. And then there was these larger family studies with inherited epilepsy syndrome. So there was these large pedigrees done where multiple family members were affected, and they started to see patterns of inheritance, thereby suggesting, certainly that there was something inherited being passed through the family. So one thing that really has emerged within the last few years in the epilepsy realm is the appreciation really of data sharing in epilepsy. So while collectively common, so the genetic causes of epilepsy as a group are quite common, but each individual underlying genetic cause, so each individual gene responsible, is quite individually rare. So in trying to explain these underlying genetic causes and underlying genetic diagnoses, the only way we're really going to reach any sort of significance is if we really start to pool these patients together. So researchers in this space have really kind of put their scientific pride aside and agreed to kind of put all of their data together. And in doing so, they started to generate these consortium. And the first was this EPI4K consortium, which just stands for Epilepsy 4000, which is a large group of scientists just pooling together a large number of patients, where they just did exome sequencing on 4,000 patients with epilepsy and really started to identify a large portion of patients with epilepsy had these what we call de novo variants, so just sporadic new variants not inherited from parents, but these new variants that arose for the first time in individuals. So from these studies, they found that genetics does play quite a significant role in epilepsy, going from single gene monogenetic causes of epilepsy to more complex inheritance. So over time, as the technology has gotten better for sequencing and different genetic technologies has emerged, the gene discovery in epilepsy has expanded quite rapidly. So we see from the early to late 90s a pretty slow progression, but as the emergence of next generation sequencing, the gene discovery in epilepsy has exploded quite rapidly. So when we think about genetic epilepsy syndromes, so each gene can be responsible for quite a broad spectrum of clinical phenotypes. So if we think about just one gene, if we use that as an example, KCNQ2 is one of our kind of prototypical epilepsy genes. It can be responsible for epilepsy across this wide phenotypic spectrum, anywhere from what we call benign neonatal seizures. So when we say benign in epilepsy, we don't mean it's any less scary than any other seizure types. So when you have a baby who's seizing in the NICU, it's terrifying to any parent. What we mean by benign is that it's usually self-resolving. Babies typically grow out of them, and usually it's in the setting of normal development. So that's really on the kind of mild end of the spectrum. Going all the way over to early onset epileptic encephalopathy, which is a pretty significant epileptic and developmental encephalopathy, to Ohtahara syndrome, which is on the most severe end, which is a pretty severe epilepsy in which babies often tend to not survive past infancy or early childhood. So that's pretty characteristic for a lot of our epilepsy genes that we see. So I won't go into too much of clinical testing, but there are a variety of testing options, and we typically employ multiple testing options to try to arrive at a genetic diagnosis for our patients in the clinic. But I think the take-home message is that currently, based on what we can offer our patients clinically, our overall diagnostic yield in epilepsy is right around 40 to 50%, which is, if you're glass half full, that's great. That's more than we were even 5 years ago. But on the other end, we're still not arriving at a diagnosis in about half of our patients. So the importance of a diagnosis in epilepsy, what's really the point? I think one of the biggest things is, really being able to provide an answer for our families. We have a lot of stories when we have families coming in with a really young baby who has epilepsy, who has these really bad seizures. There's a lot of parent guilt. It's a lot of parents, moms especially, blaming, was it something I did or didn't do during my pregnancy that caused this? So having a diagnosis really can kind of give families an explanation. So that's the one thing that we can give them. Another thing, having a genetic explanation gives the families, gives us the ability to provide some sort of prognosis for the family. We can kind of give them somewhere on a clinical spectrum that their child lies. So we can tell them, okay, this is maybe where your child is. This might be what we can expect to see in the future. Or these are some other clinical features that we typically see with this gene. These are other things we need to look for example. Some other things that we can think about are recurrence risk and thinking about additional children. So if families want to use this information for family planning, are worried about having additional children who might be affected, we can offer proper genetic counseling for them. Another thing that really emerges in epilepsy is a sense of community surrounding a genetic diagnosis. There are a lot of gene-specific family organizations that have emerged. Actually, this past weekend, I was over in California at the PCH19 conference. Melanie mentioned I run the PCH19 registry, so we were over there. It's one of the gene-specific epilepsy groups, so it's an opportunity for families of children with the same diagnosis to get together. They arrange conferences. They have their own Facebook group. They provide support for each other. So it's a way for families to get together surrounding kind of an identity and a diagnosis. One of the biggest things that is helpful, and one of the biggest things that the families really want is information surrounding treatment options, because about a third of the kiddos with epilepsy are just not responsive to medications. And if we have an underlying genetic diagnosis, sometimes it can help point to a specific treatment. So we do have specific treatments that are tailored to the underlying genetic abnormality. That's not very frequent at this point, but it is certainly an area of targeted research at this point. Okay. So that's our, really kind of our background in epilepsy. So where are our gaps in clinical diagnosis? So I mentioned that we're reaching a genetic diagnosis in about half of our patients. So where are the gaps in our knowledge? So certainly, there's variation in non-coding regions. We're doing a lot of exome sequencing. So when we do exome sequencing, that's the difference between exome and genome sequencing. We're sequencing only the coding parts of the gene. We're not doing any sequencing in the non-coding regions, so there certainly can be variation in that part that we're missing. Undiscovered gene disease associations. There are certainly genes for which we don't know their role in epilepsy and in a lot of other disorders. And also somatic mosaicism. So we talked about de novo variants or de novo mutations that arise for the first time in an individual. But these variations can also arise later, so later on in embryonic development, that can be restricted just to later tissue types, so restricted just to the brain. We're doing research in that realm as far as specifically in brain malformations, but that can also exist in epilepsy as well. So the research I'm going to talk to you about today is really going to focus on these undiscovered gene disease associations. So enter in our research-based exome sequencing. Okay. So I borrowed this slide because I wasn't sure the genetics background of the group or how much genetics overview we were going to do. But just to highlight the fact that we're sequencing and considering how any type of spelling errors in our genetic sequence can cause abnormalities in the protein, and we're considering how these amino acid substitutions or truncations or absence of these proteins can cause epilepsy, just based on the role of the protein and what it does specifically in the brain. So how about genetic variation in general? So we're looking at genetic variation that causes epilepsy, but how much genetic variation do we all have just as humans? We need to think about that kind of as a step one. So DNA itself consists of about 3 billion base pairs. So when we look at exome sequencing, so we send off exome sequencing, we get the sequencing data back. How much variation are we going to get? So when they send it off for sequencing, they compare each individual sequence to the reference. There's a reference sequence that's generated. This is the normal sequence. But what's normal, right? We're all different. We all have variation from each other. It's what makes us different as human beings. So what's different from normal? So when we get back any type of exome sequencing data, we all have about 10,000 genetic variants. That's different from what's considered normal, right? So we often send out trio sequencing. So we send out our patients' sequencing data, and we send out their parents, right? Because we want to send out as trios to kind of try to exclude what's inherited versus what's not inherited, right? To see, okay, what's unique to this family, just to exclude out which is benign variation that's just being passed through this family. But even when we do that, each individual person is going to have between 40 and 100 variants that they didn't inherit just from their mom and dad. So how do we tease out what's important from what's not important? So how do we figure out what's normal variation? So what is this single spelling base pair that's giving my patient this cute freckles, and what is causing my patient to have this catastrophic epilepsy? So first, what we want to do is to just reduce the overall number of variants that we manually want to consider, because at some point we just need to filter out some variants, and then we just need to manually go through and consider each possible variant as a cause of the patient's epilepsy. Because a lot of the times, the genes that we're looking at have never been implicated in any type of human disease, so we need to kind of figure out this on our own. So once we've reduced the number of overall variants of those that are left, we need to think about the gene and the variant, right? So does the gene make sense as an epilepsy gene? And does the variant appear to be damaging based on how that gene is supposed to function? So when we think about the variant in the protein of that amino acid, do we think it's going to impact the structure of the protein? Okay, so step one. So we get back our exome sequencing data. It's going to look like a huge Excel spreadsheet with about 10,000 rows. So we set our filters. And the programs that we use are actually really cool because they import some data that we can kind of manipulate. So we can set our filters in two ways. We can set them based on expected inheritance patterns. So we have the data with our patient and with our two parents, usually, if we're lucky. We have our parents, sometimes we don't, and it's a horse of a different color. We won't talk about that for now. But we can set our filters based on inheritance. So we can set a de novo filter. So what variants did our patient not inherit from parents, which is a good quick first pass just to see, because we talked about earlier that a lot of our epilepsy genes are de novo. So that's a good first filter to use. We can set autosomal recessive filters. So we can say, okay, what's a gene that they inherited from mom and dad that the patient has 2 copies in this gene that mom and dad are both carriers of. We can do the same thing if we have a male patient. What did they inherit from mom on the X chromosome? Because males have 1 X chromosome, they're typically affected when moms are unaffected carriers. So we can use those filters. Another filter that we use is these population filters. So I'll talk about Nomad later. Guyun alluded to it earlier, but it's this great resource that's publicly available now. It basically is a public resource that provides provides exome sequencing for about 120,000 presumably healthy individuals. So it's a good filter that we can use to filter out variants that are common in the healthy population. So we can whittle it down to something more manageable. So we start with 10,000, which is daunting. After our filters, we can get it down to about 40 to 50, which is kind of like, all right, that's kind of a lot still. Then I do something really scientific called like the eyeball check. I just do a quick scan and quickly get rid of some more, just based on the type of variant, gene class, and just get down to about 10 that I wanted to start to manually curate, which makes me happy as a good first step. Okay, so then we start to, remember I said after step 1 we do the gene level and then we go to the variant level evidence. So what do we know about the gene? So we start with just a literature search. What do we know about this gene so far? And this is probably where you guys have the most expertise. We do a quick lit search. PubMed is a good start. I actually usually start with OMIM, so Online Mendelian Inheritance of Man. It's a good resource that kind of compiles a lot of information that is known about a particular gene. It's a good search for, it's a good starting point. You can just type in your gene, and it kind of compiles everything, and it has the links out, PubMed links for all of the resources. Oftentimes, because there are no disease association with these genes, there's very little information. At most, we might get like a mouse model, but rarely we'll actually ever like get anything. So a good next step is if a gene has never been implicated in any particular condition, there are resources available that will tell us where a gene is expressed. So we can look at the expression pattern. So as a fun fact, so the DNA in all of our cells is pretty much the same, but we're not all walking around as kind of a big blob of undifferentiated cells, right? Our bodies are unique networks. We have different organ systems that function each in individual ways. And that's because of gene expression. So our genes in each part of our body are expressed in different ways. So we can use that information when we're trying to think about what a gene does. Okay? Here's an example. GABRG2. So if we find this gene, we don't know what it does, but we want to think about, okay, where is this gene expressed? We can use this online tool, it's called GTEx, the genotype tissue expression. We can type it in. I don't know if you guys can see this here, but this has all different tissue types. So we can see where we get a hit, where all of that yellow is right there. And if we look here, all this hit is all in brain. So we can say, okay, so this gene is expressed in brain. If you have an abnormality in this gene, we can infer that we're probably going to end up with some sort of neurological phenotype. So that's a good kind of starting point. So if my patient has epilepsy, and there's an abnormality in this gene, I would be like, okay, I'm highly suspicious that this could be implicated. So this actually is a GABA receptor, so this is a known epilepsy gene. It's not always this simple. So this, I got this result actually on a clinical test for one of my patients who had epilepsy and autism. So here's our brain section. But as you can see, this gene is expressed everywhere as well. So this came up as a candidate gene on a clinical exome test. Turns out a couple months later, it was implicated in a handful of other genes with epilepsy as well. So we can't let this fool us either, because this turned out to be another epilepsy gene without any other extra neurological features. So it can be tricky. All right. So more kind of gene variant level information. So I'm going to go into EXAC and NOMAD. So I'll skip this for the sake of time and go back to it. So NOMAD stands for this Genome Aggregation Database. So this is an online, publicly available database where they pulled sequencing data from a variety of sources, and they publish it all online. So you can search it by gene, and it pulls up all of the variants that were reported and the number of individuals who had each of those variants. So here on the left, we can see the protein change. So this is all the proteins in the position number. The type of change, so missense just means that it's a single amino acid substitution. And it just shows the number of people, number of individuals who had that variation. And the allele frequency over here. So this is helpful. So our first step where we did the filters, where we can filter out based on population. So we filtered out based on a certain allele frequency, but it's not going to make it so that these numbers are going to be 0. There's still going to be a couple people that could potentially have it. So we still want to check this, even though a gene popped up or a variant popped up on our list. So we're going to cut it off based on high populations, but we still expect to see a couple. So we do want to check to see the number of people in NOMAD that do have this. I mean, I'll go back up, and this is some helpful information too. So EXAC was kind of an earlier version of NOMAD. So EXAC was the Exome Aggregation Consortium. So it had 60,000 exomes, whereas NOMAD doubled the number of exomes and added some genomes. But what's still helpful about EXAC is it has these constraint measures. So somebody went through who was really smart and did some math, and they were able to tell that based on the size of the gene and the makeup of the gene, they're able to tell the number of expected variants you would expect to see just based on the makeup of the gene. And then they're able to go through and say, okay, the number of healthy people who have variants in this gene, based on their sequencing data. So they take the expected number of variants and the observed number of variants, and they're able to calculate how tolerant a gene is to variation. And they're able to generate these z-scores, okay? So if a gene is not tolerant to variation, they're going to have a higher z-score, okay? So for ROBTB2, we're not seeing as many observed variants as you would expect to see, so that gene is not tolerant to variation because there are just not as many in the healthy population. Versus MBD5, you're actually seeing more variants than you would expect to see, so it has a very low, actually a negative z-score. So we kind of use that information too, so if you see a missense variant in MBD5, you're not really going to be super concerned about that, but if you see one in this gene ROBTB2, then you'd be a little bit more concerned about that. So that's just another piece of information we use. In silico prediction scores, these are just computer models that we use. It's kind of like lower on our kind of scale of evidence that we use, but basically these are computer models that are able to use algorithms that think about the three-dimensional protein structure and the effect of an amino acid on the overall structure. And they churn out a score from kind of 0 to 1, from benign to damaging. And they also use evolutionary conservation. So they look at different animals over evolutionary time, and they look at whether or not a particular amino acid position is conserved over time. Let's see. And then some variant level evidence. So was a particular variant itself previously published? This we use more on a clinical basis rather than on a research basis because typically our genes haven't been really established in any particular disorder, so oftentimes they're not published on particular variants, but it's good to check. So this is just a helpful clinical resource. So this is HGMD, so this is the Human Gene Mutation Database. This is not publicly available. You need to have a subscription for this, but basically you can plug in any gene of interest, and they'll curate all of the variants that have been published in the literature, and then they will link out to all of the primary literature that published that variant. So they'll have the first publication, and then usually maybe a second or third publication that also reference that same variant, but usually only the first couple. So that's a helpful resource as well. So wrapping up, so we kind of go through this process for a lot of our variants, but still at the end of the day, it's very limited with what we have. So we do have tools. We just end up crowdsourcing. That's very big nowadays, right? Just crowdsourcing for things. So Gene Matcher is something that's relatively new. It's a way that we can kind of connect with other researchers and clinicians who may have variants in the same gene. And the way that it works, so say this is me in Boston. I have a patient with a variant in gene ABC who has epilepsy, developmental delay, and hypotonia. This is a geneticist over in California. He also has a patient with a variant in gene ABC. His patient has dysmorphic features, epilepsy, and developmental delay. There's no way we would ever know about each other. So we both put our information into this online Gene Matcher, and then Gene Matcher says, hey, there's a match here, and notifies both of us. At that point, we know each other exists. We can connect and say, hey, I have this patient. He says, hey, I have this patient, and then we can compare notes and then make a connection, which is great. So sometimes we're able to find an answer for our patients, but we might not be able to do that right away, and sometimes only time will tell. The information surrounding the genetic basis of epilepsy is really expanding over time, so sometimes it requires just continual reanalysis every few months and just revisiting the exome data over time. We do that on our end. There are a variety of other research projects who do that as well. The Epilepsy Genetics Initiative is another project that does that on a clinical, well, it's a research basis, but it takes in clinical exomes, and they enroll patients who've had clinical exomes who have epilepsy, and they reanalyze it every 6 months. So we've been working with them, and we also advise patients who've had clinical exomes to get their exomes reanalyzed every year or so, just because the information is changing so much over time, and we're learning so much more. So just to wrap things up, you know, many of the early onset epilepsies do have a genetic etiology, whether or not we can find it. In 2018, we do think that it's there. You know, we've come a long way, but we certainly do still have a lot to learn, and it's, we do have a lot of ongoing collaborative efforts, and hopefully we'll continue our upward trajectory. So we have a lot of cool tools at our disposal. I've showed some of them to you today. So little pieces of evidence, you know, they do add up. So hopefully we'll continue to make advances and have more resources and tools that we can use. So with that, I'd like to thank our team, our epilepsy genetics program at Boston Children's Hospital, and patients and families who, you know, have taken this journey with us, who trust us for our clinical and research care. And as we transition to our Q&A, I'll leave up some helpful resources. Thank you for your time.
All right, we do have time for Q&A. Now, we do have a little bit of question with a microphone. So these two microphones can't be too near each other. If you wanted to, here, I'll take this one question.
Hi. I have two first cousins that died of breast cancer, so 4 years ago I wanted to have BRCA testing, and insurance didn't cover it, so $4,000. But I waited, and so now I did it this year, and it was covered. So how is insurance covering all of this cost?
Sure. I can speak to the cancer genetic testing aspect. Certainly, as technology improves, costs go down, and as costs go down, the cost burden as perceived by insurers go down as well. But the other part of it is that clinicians, we have made so much effort into pooling our resources and forming practice guidelines within consortiums that insurers pay attention. So Medicare, if Medicare pays attention to BRCA testing and covers BRCA testing, more insurers fall in line. The other aspect with BRCA testing in particular is competition. And so now more labs have provided genetic testing. It used to be was due to gene patenting issues, could only be provided by one lab. But in 2013, the Supreme Court ruled in favor of the ACLU that put up a case saying that gene and genetic technology should not be patented, and allowing for more testing companies to come into the genetic testing market, particularly for BRCA1 and BRCA2 genes. So that's just a little bit of comment on that. But genetic testing really does run the gamut of the life cycle, and genetic testing coverage overall has been evolving due to cost of the technology going down as well. Any additional comments? Do you want to comment on clinical exome?
I think insurance coverage, you know, it really varies based on specialty. I think a lot of the pushback we get in neurology is there's really limited evidence on how it's going to impact clinical management. I think in cancer, there's really a lot more literature out there of how a genetic test is going to impact management, because you can say if you have a genetic predisposition, it's going to increase screening, it's going to, you're going to do all of these things differently. In epilepsy, there are so few instances where it's going to change management, so a lot of insurance companies are saying, oh, genetic testing in epilepsy is considered investigational, it's considered experimental, so a lot of insurance companies just flat out are denying it, aren't covering it. So we're kind of pushing now on trying to get research out there and trying to get data to support ways in which management will change for neurological conditions. So that's kind of where the efforts are now. But it certainly is changing, it's evolving as new information becomes available.
Thank you. I really appreciate it, especially in all the talks talking about sort of like consent and informed consent. And I was wondering if you could talk a bit about data privacy, especially for people who have like rarer genetic conditions, people who are kids or teens, with these sites like GeneMatcher or the case studies in the literature. How is that handled in the field?
So a lot of the data shared is shared in a de-identified way as much as possible. You know, the extent to which you can share clinical information without being too identifiable, without sharing, you know, obviously name and date of birth, but really information on specifics of age of onset of certain things and things like that. But as far as information getting out there, it's, I don't know, I mean, is there anything else to add?
Just to add in terms of like something like GeneMatcher and some of the other crowd sharing databases like Face2Gene, so you can input data without identifying the person themselves. So it is a limited data set, and it's really motivated by, in the case of Face2Gene, phenotypic features. However, we do require consent for something like Face2Gene, so the parents have to consent to allow a photograph to be uploaded. When it comes to GeneMatcher, it is a limited data set, so doesn't have identifying information. In the clinical genetic testing arena, I think that is a huge concern and a very valid concern. When it comes to reporting a case report, I mean, that's very identifying. So we only do it with parental and the individual's consent when appropriate. So in the case of the minor who had genetic testing that I presented, we had consent from her and shared a copy of the case report with her.
And I guess at the utmost extreme, working in a Huntington's disease clinic, where it can be quite burdensome for someone to consider testing for Huntington's at an age where they're not showing any symptoms. Someone who tests positive for Huntington's disease has a 100% chance that they will show symptoms someday unless we successfully develop a treatment before that time. So many patients who come to my clinic wanting to have predictive testing will elect to do that testing outside of the medical record and pay out of pocket. We have gone through many hoops within the hospital to allow us to keep paper records rather than in the hospital. The Genetic Information Non-Discrimination Act does prevent your employer and health insurance from discriminating against you based upon your genetic information in the majority of cases, but it's a little hard to prove that genetic discrimination happens and exists, which is why some individuals elect to go through testing in that way despite those protections.
Okay, just a quick, I don't have a question. I'm making a quick public service announcement. Just as a reminder, as the Q&A is happening, we do have our evaluations for each session online. You can get to them by going to the Science Bootcamp LibGuide. So if you Google, if you don't have it bookmarked, Google New England Science Bootcamp 2018. I think Melanie might be putting the slide up that shows the site. So that's a Google form, and then make sure you get your button on your way out.
And don't forget to evaluate yesterday's session also, which we didn't specifically remind you to do.
I'll have the link for that as well.
Hi, so kind of following up on this genetic privacy question.
right now, we use debit cards, credit cards, that puts information out there that can become hackable information. Amazon, Google watches what we look at, what we buy. They use that to market to us. Do you guys have any concerns about just genetics sort of falling victim to consumer marketing and or people wanting to target genetic information? I know I'm asking you to see in the future, but I mean, I'm just curious.
That's a really...
I'm thinking you all think about this stuff, so I'm just kind of curious.
I think that illustrates that we've done a good job giving you a sense of the field, because I think as genetic testing has become more accessible, especially sort of by stepping a medical provider, I think a legitimate concern of some of these direct-to-consumer companies. And so part of how these companies are making a profit is that the public is curious about their own genetics and then decides to send in a DNA sample to, learn if their child is good at sports, which we cannot do yet, or their ancestry, which we're better at doing. But a lot of how these companies actually continue to survive is then they take their data sets and sell them to other individuals. And there's been, I don't think we're there yet in terms of because we don't know enough about our genome altogether to really look at a genome and find that person. But I think with time, I think we'll be better at doing that. So there has been a few things published where they kind of talk about how it actually is possible to take someone's genome and then actually figure out who that person is. So I think I did 23andMe because a lot of my patients were coming into clinic telling me about it, and I felt not equipped to really understand what they were told and what I should believe about it. And in retrospect, I wish I had maybe done it under a pseudonym or something like that, just because you don't really know where time will go in terms of data. And I think, we don't really know, there was some talk over the current presidency about GINA and health insurance and all this. So the current protections that exist were passed through bipartisan efforts. We hope they will continue to stay strong, but time will tell how everything will evolve. But I think that is definitely a legit concern as more information is put out there. Do you guys, that's a loaded question. Do you have anything to add?
Hello. One of the genetic counselors in the video that you showed mentioned genetic counseling as personalized medicine. And I was wondering if you could comment more on that movement towards personalized precision medicine and the role that genetic counseling is playing there.
Sure. I mean, Lacey and Guy probably have more examples of what's happening right now, but the clinical research study that I'm starting up on relating to Alzheimer's disease is really exciting because it's basically taking someone's genetic information and other physical risk factors and then hopefully intervening with medication to ultimately prevent symptoms of Alzheimer's disease. So in other diseases, I think we've made even more progress, which these two ladies can allude to.
Quick answer in the oncology realm. Certainly when it comes to treatment decision making, particularly in patients who have metastatic cancer, genetic testing has definitely entered the treatment decision making realm. Examples are in a BRCA1 or BRCA2 gene mutation carrier who has metastatic ovarian cancer, then they can take olaparib as a agent that would target their ovarian cancer cells, their BRCA1 or BRCA2 deficient ovarian cancer cells. Also in the realm of lung cancer and Tarceva treatment with EGFR mutations that can be specific either somatically within the lung cancer itself or in very rare cases can be in the germline. But that is actually another example of kind of treatment decision making with genetic testing on specifically on the lung cancer itself.
In epilepsy, this has really been an area of focus. So it can be on various levels. So for specific genes, there are certain drugs that certain classes of drugs that are known to work on specific genes. So for SCN1A, for example, we know to avoid sodium channel blockers. So if a patient has an SCN1A variant, we know to avoid that class of medication. Ezogabine works particularly well for patients with KCN2Q variants. And even on a more kind of sci-fi level, we're involved in a research study now that for patients with KCN2Q2 variants, we're taking some blood samples or some skin samples. We're growing them in a dish. We're turning them into stem cells, and then we're going to screen them for a variety of different compounds to find out which drug works best, because we're turning their skin cells into neurons, and we're going to see how they fire, and then we're going to see which drugs work on those particular neurons based on not only their own genetic variant, but how their genetic variant is working in their own genetic background. And then we're going to hopefully translate that back into their treatment. So it can be on various levels. So it sounds kind of sci-fi, but that's certainly where we're going.
Because certainly a brain biopsy is definitely far more traumatic than a skin biopsy, right?
Yes. We try not to take our patients' brains when we don't have to.
This might not be a question you can answer, but I was still wondering about it. Kind of piggybacking off of the insurance coverage issue that was mentioned before, I was wondering if your field is concerned or talking about disparities in healthcare influencing the information that you have available in the long term and how that influences care going forward for those populations.
Sure. So prior to coming to Brandeis last year, I was at Boston University School of Medicine for about 5 years, which is a safety net hospital and serves a very diverse patient population, and had worked in some other community hospitals in Massachusetts before that, and became passionate about those issues. So it's hopefully improving over time, but a big problem you encounter with patients who either have a lower socioeconomic status or are of racial minorities is first, they may have insurance plans that are not covering the testing, but that's not always true. Sometimes Mass Health is actually better than some of the other insurance companies, ironically. But then when you go to search some of these databases, it's more common that the individuals in the database are white individuals, and so it was definitely a struggle at Boston Medical Center when we did genetic testing, and they were just seemed more likely to have variants that were more difficult to rule if they were pathogenic or benign, and some of that just had to do with lack of data on their population. As a field, as a whole, perhaps similar to librarians, we are white and Caucasian, the vast majority. So in terms of providing appropriate care towards all of our patients we work with, we really are trying to make more efforts in making our profession more similarly match the population of the United States, which I think is an issue that many healthcare professions are facing, but it's definitely true in genetic counseling.
One last question. No, you got it.
To continue to the sci-fi side of this process, what is the relation between the protein data banks and so on? Is there any way to analyze proteins to find out, okay, this person has that type of genetic disease and so on? So is this soon, or is this sci-fi side, or is there any protein aspect in this analysis?
Yeah, so actually, like, proteins can be enzymes, and so some of the genetic disorders that we understood first as a profession were metabolic disorders that were easier to test for us through like newborn screening. So, for example, PKU is a fairly well-known metabolic condition where your body is not breaking down certain amino acids well. You can get evidence of that by looking for that enzyme function in the blood. So, so there are a fair number of conditions out there that you can screen for them by looking for proteins in either the urine or the blood. A lot of these are metabolic conditions. But part of genetics that we're kind of getting there and understanding the genes, but a really exciting area of genetics is epigenetics. So that doesn't have to do with the spelling of the genes, but which genes are turned off and on at certain times. And you know, some of that has to do with what proteins are present or absent. So I think there's multiple layers to understanding why diseases happen. For example, autism is extraordinarily common right now. Even with the best genetic testing out there, we're still only able to find a cause in less than half of kids with autism. So the current thinking is that maybe there's other factors, either having to do with proteins or epigenetics, that explain why that happens. So maybe in a decade we'll have more information for you.
It's on. Thank you so very much. This was a wonderful session. Round of applause. All right, you're off to lunch at the faculty club. We're back here at 1:30 for material science. And